Mutations come in all different forms that occur during cell division processes in our bodies. Let's look at what a normal cell cycle looks like. Here we can see that the cell cycle is composed of interphase, mitosis, and cytokinesis. These steps are regulated by specific checkpoints. Within interphase, these checkpoints include G1 and G2. Then the M phase has a checkpoint as well. In this way, the cell cycle is like a washing machine. Internal and external signals help the cell continue or stop its cycle. Cyclins are proteins that cyclically fluctuate in concentration, and they play an important role in regulating the cell cycle. By accumulating in the late S phase, then combining with other proteins, cyclins promote mitosis while regulating the rate at which it occurs. But what happens when the cell cycle isn't regulated? Now it's time to get into mutations. Mutations are changes in the genetic material of a cell or virus. Two types of mutations are DNA and chromosomal mutations. Let's talk about DNA mutations first. Point mutations occur when a base pair of a single nucleotide in a DNA strand is changed. Point mutations can be divided into two categories: base pair substitutions and base pair insertions or deletions. Base pair substitutions, which simply replace one pair of nucleotides with another, can be further divided into three categories: silent, missense, and nonsense. In a silent mutation, a change occurs in the DNA and mRNA sequence. However, the mRNA still codes for the same amino acid. Missense is when one amino acid changes. Nonsense is when a stop codon ends the chain. Missense and nonsense can also be considered to be frameshift mutations, which fall under the categories of insertion and deletion. In insertion, a nucleotide is added to the sequence. In deletion, a nucleotide is removed from the sequence. Not all mutations are bad, but some can cause detrimental impacts, such as cancer. Mutagens, agents that cause mutations in proto-oncogenes, resulting in oncogenes, cancer-causing cells. When a gene is mutated to cause cancer, the genes cause cells to rapidly divide, ignoring the checkpoints that we talked about earlier. Other genes that can be mutated to cause cancer are the RAS and P53 genes. Enough about DNA mutations. Let's talk about chromosomal mutations. If a pair of homologous chromosomes do not separate normally during meiosis, a mutation may result due to the non-disjunction. If a gamete is fertilized yet has been a victim of non-disjunction, an aneuploidy comes about, which means that the zygote has an abnormal number of a particular chromosome. The zygote could end up being monosomic, having only one copy of a certain chromosome. It could be trisomic, having 3 copies of a certain chromosome, or it could simply be polyploidy, having more than 2 complete sets of chromosomes. Mutations, they are everywhere.